Good morning, everyone. Today, we embark on a journey to demystify the intricacy of quasi exam questions for AAS level students, examining the post offer us to compel us to navigate the challenges ahead, empowering us with insight and strategies to conquer the present. Let's delve into the treasure trove of quest exam, unlocking the key to success and mastering the art of academic excellence. Until recently, the typical viruses known to science were 20 to 150 in size. In 2003, the mini virus was discovered with a size of approximately 680. In 2013, the Pandora virus was discovered, which has a size of over 1000 millimeter. Which viruses can be seen using a light microscope with a maximum resolutions of 0.25 and an electron microscope. With a maximum resolution of 0.25 micrometer, light microscope can effectively visualize viruses such as typical viruses ranging from 20 to 150 nanometer size, as well as the mini virus discovered in 2003 with a size of approximately 680. Electron microscopes, on the other hand, with a much higher resolution, can visualize viruses of smaller sizes, including the Mimi virus and Pandora virus. Therefore, both the Mimi virus and Pandora virus can be visualized using an electron microscope. The answer will be typical viruses cannot be seen. Mimi virus and Pandora virus can be seen. The diagram shows an eyepiece graticle and a cell view through a microscope. When the eyepiece graticle was calibrated at this magnification, the whole length of the graticle shown covered 12 divisions of a stage micrometer scale. There were 100 divisions in 10 mm of a stage micrometer. What is the actual length of the cells? It is 360. Tay-Sachs disease results in a build-up of lipid in cells, which cell structure does not function correctly in this disease. Tay-Sachs disease primarily affects the lysosome within cells. Lysosomes are responsible for breaking down various substances, including lipids. In Tay-Sachs disease, the deficiency of the enzyme hexosaminidase a result in the accumulation of lipid called GM2 ganglioside within the lysosome, leading to a cellular dysfunction and ultimately neurodegenerations. The answer will be lysosome. Which animal cell would have the most extensive Golgi bodies? Ciliated epithelial cells would likely have the most extensive Golgi bodies because in these cells line various organs such as the respiratory tract and the fallopian tubes. They are involved in the secretion of mucus and the coordination of ciliary movement. And these cells require extensive Golgi bodies for the productions and secretion of mucus, as well as for the synthesis and assembly of ciliary components. The diagram shows three circles, 1, 3, and 5, and the shared structure 2 and 4. Which row correctly identify the three circles and some of the structures that are shared between them? Circle 1, circle 2, circle 3, 4, and 5. So, prokaryotes are single cells organism that lack a nucleus and membrane-bound organelle. They typically have circular DNA, which is found in a nuclear region, and mitochondria and chloroplasts are organelle found in eukaryotic cells, and they are believed to have originated from prokaryote placenta through endosymbiosis, and both mitochondria and chloroplasts have their own cellular DNA, similar to prokaryotes, and replicate independently within the eukaryotic cells. And the answer will be C. Which row about the genetic material in animal cells and prokaryotic cells is correct? And animal cells do contain linear DNA, which is found within the nucleus. However, prokaryotic cells such as bacteria also contain double-stranded DNA, but it is typically cellular and found in the nucleate region. So while both contain double-stranded DNA, prokaryotic cells have cellular DNA. And prokaryotic genetic material is not surrounded by a double membrane. Instead, it is typically found in the nuclear region without a membrane. And the answer would be animal cells contain linear DNA is correct. Prokaryotic genetic material is surrounded by a double membrane, and this is not correct. And prokaryotic genetic material is a double-stranded DNA is correct. What is the present in all viruses? Guanine. The test for non-reducing sugar require a second Benedict test to be carried out. Which set of tests is the correct method for carrying out the non-reducing sugar test before carrying out the second Benedict test? First, perform the Benedict test. Then, if the solution remains blue, indicating the absence of reducing sugars, then can proceed with boiling the solution with dilute hydrochloric acid to hydrolyze any non-reducing sugar present. After that, neutralize the solution with sodium hydrogen carbonate before performing the Benedict test again. This will allow to check for the presence of reducing sugar after hydrolyzing the non-reducing sugar. And the answer will be perform the Benedict test with the color of the solution remaining blue, boil with dilute hydrochloric acid, and neutralize with sodium hydrogen carbonate. The diagram shows three types of covalent bond. Which bond will be found in glycolipid and glycoprotein? The answer will be bond 1 represents the glycolipids only. Bond 2 is a glycolipids and glycoprotein. And bond 3 is a glycoproteins only. Which molecule would be found in an oil liquid more than in fats liquid? If you look at the original structure, lipids consist of oil and the fat, but the oil is actually consists of double bond, and fat is only single bond. From this diagram, from diagram 1, 2, 3, and 4, which molecule diagram represent double bond? Only 2 and 4. 2 and 4 is actually an oil. The answer will be 2 and 4. Which row about the bond found in the primary, secondary, tertiary, and quaternary structures of protein is correct? The answer will be primary is a covalent, secondary is hydrogen, tertiary and quaternary is a hydrogen, ionic, covalent, and hydrophobic interaction. The specific heat capacity of different substances is shown. Which statement is correct? It takes more energy to raise the temperature of hydrogen gas than it does to raise the temperature of water. So the answer will be air is a more stable environment than water because it is more resistant to changes in temperature. The solid line of the graph represents the product forms over time for a reaction in a cell, which other line represent the effect of aiding the enzyme for this reaction. The answer will be in. The graph shows how the rate of an enzyme control reaction is affected by the change in temperature. All other variables were standardized. And which is the factor limiting the rate in region X? It is temperature. How will the removal of a reversal non-competitive enzyme inhibitor affect an enzyme catalyst reaction? The removal of reversible non-competitive enzyme inhibitors typically result in an increase in enzyme activity. And since non-competitive inhibitors bind to allosteric sites rather than the active site, removing them allows the enzyme to function at its maximum potential, leading to increased catalytic activity and potentially higher reaction rates. And Km represents the Michel's constant, which is a measure of the substrate concentration at which enzyme catalyzes the reaction at half its maximum velocity. And then the answer will be C. The dimension of three agar cylinders X, Y, and Z are summarized in the table. And to calculate the surface area to volume ratio SA over V for each cylinder, we first need to compute the surface area and volume using the formula. And then for the cylinder X, the surface area over the volume ratio, we get the value of 1.2. And for cylinder Y, the surface area over the volume ratio, we get the value of 2.1. And for cylinder Z, to get the surface area over the volume ratio, we get 0.8. So the correct order of the surface area to volume ratio from smallest to largest will be Z, smaller than X, smaller than Y. The answer will be C. Three individual plants leaf cells were each placed in a different solution for 30 minutes, and each solution has a different water potential, which will correctly show the change in volume of the three cells after 30 minutes. When the water potentials of the solution is lower than of the cell will lose water and shrink in volume. When the water potential of the solution is equal to that of the cells, there will be no net in volume. So when the water potential of the solution is higher than that of the cell will gain water and swell in volume. So the changes in volume occur due to the movement of water molecule across the cell's membrane through osmosis. And water moves from area of higher water potential to area of lower water potential. So the direction or magnitude of the volume change depend on the relative water potential of the cells and the solutions. The answer will be B. Which root is a representation of one chromosome at the beginning of prophase of mitosis and the number of DNA strand in the chromosome? The answer will be 4. Number 19, which event are part of mitosis? Interphase, telophase, or cytokinesis? Telophase is one of the phase in mitosis, and it is the final stage before cytokinesis, where the chromosome reach opposite poles of the cell and begin to decondense. So the answer will be 2 only, telophase. The statements are about gene and protein involved in breast cancer. The protein coded by the BRCA1 gene inhibits the growth of breast cancer cells, and the protein coded by the p53 gene suppresses tumor. Which combination of gene is most likely to result in breast cancer? So the answer will be A. The combination most likely to result in breast cancer in when both statements are false. If BRCA inhibits cancer cells growth and p53 suppresses tumor, so the absence or mutation of one gene would increase the risk of breast cancer. The photomicrograph shows a cells during mitosis. What is happening in these cells? Chromosomes are condensing, centromeres are moving to the opposite poles, and the spindle microtubes are shortening. The answer will be centromeres are moving to the opposite poles, and spindle microtubes are shortening. Which diagram correctly shows the structure of a piece of DNA? A piece of DNA typically consists of two strands that twist around each other in a double helix structure, and each strand is made up of nucleotides, which are composed of a superphosphate backbone and nitrogenous bases. The nitrogenous bases, adenine, thymine, cytosine, and guanine, pair up with each other across the strand. Adenine with thymine, if you can see, and cytosine with guanine, forming complementary base pair. And this structure allows DNA to carry genetic information in the sequence of its bases. Bacteria were grown in a medium containing N15 after several generations all of the bacteria DNA contained N15. Some of these bacteria were transferred to a medium containing the common isotope of nitrogen, N14. The bacteria were allowed to divide once. The DNA of some of those bacteria was extracted and analyzed. This DNA was all hybrid DNA containing equal amount of N14 and N15. The remaining bacteria were left in a medium with N14 and allowed to divide one more time. The DNA of some of these bacteria was extracted and analyzed. What is the percentage of hybrid DNA? After one round of division in the N14 medium, the DNA of the bacteria become hybrid, containing equal amount of N15 and N15, N14 and N15. After another round of division in N14 medium, the DNA would be a mixture of hybrid DNA and DNA containing only 14N. In the second round of division, half of the bacteria would have undergone replication, resulting in DNA with only 14N, while the other half would retain the hybrid DNA containing equal amount of N14 and N15. So, after the second round of division, the percentage of hybrid DNA would be 50%. The answer will be B. Which statement correctly describe the process of transcription? The answer will be only 2. A section of DNA is converted to RNA by RNA polymerase. Number 25, sickle cell anemia is caused by a mutation in allele of the gene that codes for the beta globin, polypeptide, and hemoglobin. The diagram shows the sequence of bases in a small section of a template strand of DNA for both HbA normal and HbS sickle cells beta globin alleles. Both the polypeptides for HbA and HbS are the same length. How will the mutation in the allele result in the production of an altered version of the beta globin polypeptide? In the case of the HbS allele where the codon GAG is mutated to GTG, a tRNA molecule with the anticodon GUG would indeed form hydrogen bond with the altered codon of mRNA. This tRNA molecule would carry the amino acid valine, which is specified by the codon GTG. Therefore, during translation, the tRNA with the anticodon GUG would correctly recognize and bind to the mutated mRNA codon, allowing for the incorporation of a line into the growing polypeptide change. The answer will be a tRNA molecule with the anticodon GUG will form hydrogen bond with the altered codon on mRNA. So the plan diagram of the transverse section through a dicotyledonous plant stem was drawn by a student. They have stained the sections with a chemical that stained lignin. Which row is correct for tissue? W and Z. W is actually a represent folium, and Z is actually represent xylem, and the stained tissue would be Z. Number 27, the question is discounted. So whatever that you answer will be giving the right for you. 28, a plant leaf scenes in transverse section with a microscope show the features listed. A thickly waxy cuticle on upper surface, sunken stomata on the lower surface, and wool leaves to the edge firm. What is this leaf adapted for? The leaf is likely adapted for reducing water loss through transpiration. The thickly waxy cuticle on the upper surface helps to prevent water loss through evaporation, while the sunken stomata on the lower surface reduce water loss by creating the microenvironment that traps moisture. The rolled edges further minimize water loss by reducing the exposed surface area for evaporation. And these combinations of features is typical of plant adapted to arid or dry environment. So the answer is to decrease water loss by transpiration. What occurs as carbohydrate is taken out of a sieve into a folium sieve tube element. So when carbohydrates are taken out of a sieve into a folium sieve tube element, the water potential in the folium sieve tube element would decrease due to the increase in solute concentration. And this decrease in water potential would cause water to move into the volume sieve tube element from surrounding tissues, increasing its volumes. Conversely, when carbohydrates are unloaded from the volume sieve tube element at the same, the water potential would increase, which leading to water movement out of the volume sieve tube element and the decrease in its volume. The answer will be when the water potential in volume sieve tube element becomes lower, volume of liquid in volume sieve tube element is increasing. Which factors affect blood pressure? The diameter of the blood vessel is one of the factors. Blood pressure is influenced by the diameter of the blood vessel. Narrowing of the vessel called vasoconstriction increases blood pressure, while widening called vasodilation decreases it. The systolic pressures on the heart ventricle refer to the pressure exerted by the heart ventricle when they contract and push blood into the arteries. The higher systolic pressure correlates with the higher blood pressure. And the volume of blood returning to the heart in each cardiac cycles mean that this is known as a preload. An increase in preload due to factors like increased renal return leads to increased blood volume in the ventricle, which can elevate blood pressure. So the answer will be 1, 2, and 3. Sutemi is a module that insipid carbonic hydroxide, which effect would sutemi have inside the red blood cells in muscles tissue that is respiring at the high rate. Sutemi is a bicarbonate hydroxide, an enzyme involved in the conversion of carbon dioxide to bicarbonate ion, which is crucial for buffering pH changes in the red blood cell. In muscle tissue, respiring at the higher rate, there is an increased production of carbon dioxide due to increased metabolism. If the formation of hemoglobinic acid is decreased, it could lead to a decrease in the buffering capacity of the red blood cell, which is affect the ability to transport carbon dioxide efficiently. This might result in an altered acid-base balance within the red blood cells, impacting their function in gas exchange and potentially affecting overall oxygen delivery to the tissue. So the answer for this will be a decrease in the rate of formation of hemoglobinic acid. Number 32, the partial pressure of oxygen in the blood vessel in different parts of the body affect the percentage saturation of each hemoglobin molecule with oxygen, which statement explains the difference in saturation between a resting and an active muscles. A small decrease in partial pressure between K and 7 causes 2 molecules of oxygen to leave each hemoglobin molecule because the oxygen dissociation curve is steep. Which statement about the Bohr shift are correct? 1. The shift is accelerated by the action of carbonic hydroxide. 2. A decrease in blood pH will cause oxyhemoglobin to dissociate. And 3. A decrease in carbon dioxide concentration will cause more oxygen to bind to hemoglobin. Statement 2 is correct because the Bohr shift refers to the phenomenon where a decrease in blood pH and increase in acidity, which is causes oxyhemoglobin to release more oxygen to the tissue. Statement 1 and 3 is considered incorrect. Statement 1, because carbonate hydroxide catalyzes the conversion of carbon dioxide and water to bicarbonate ions and protein, but it doesn't directly affect the Bohr shift. Statement 3 is considered incorrect because the Bohr effect states that an increase in carbon dioxide concentration will cause more oxygen to be released from hemoglobin, not the other way around. Some of the events during the cardiac cycles are listed. First, left and right arterial contract. 2, left and right ventricle contract. 3, a wave of electrical activities is conducted by the kidney tissue. And 4, a wave of electrical activity passes through the atrioventricular nodes. What is the correct sequence of events after the cerebral node sends out a wave of electrical activities? The answer would be not on this, is either A, B, C, or D. The correct answer would be a wave of electrical activity is conducted by the activity tissue, and then left and right atria contract. Next, it will go to a wave of electrical activity passes through the atrioventricular node, and finally they have left and right ventricle contract. Number 35, the question is also discounted. And number 36, which statement is correct? Cholera is caused by the virus Vibrio cholera. Mosquitoes are pathogen that cause malaria. Microbacterium bovis only causes TB in women, and Plasmodium vivae is example of pertussis. The statement D is correct. Plasmodium vivae is indeed an example of protozoa or pertussis parasite that causes malaria. Cholera is caused by the bacterium Vibrio cholera, but not a virus. And mosquitoes are vectors for the transmission of malaria, but they are not pathogen for themselves. And Microbacterium bovis can cause TB, tuberculosis in human, but it's not limited to human, but it can also infect other men. So the answer will be D. 37. The graph shows the changes in the antibiotic resistance of a specific bacterium between 2000 and 2016 in one country. Samples of bacteria were collected every year from 48 hospitals, and the bacteria were tested to see if they show resistance to 5 different antibiotics. And what can be concluded from the data in the graph? Percentage resistance to 3 of the antibiotic was at peak in 2015. 38. Some responses made by cells of the immune system to the pathogen are listed. Mitosis, bind to specific antigen, produce memory cells, or secrete antibodies. Which responses are correct for B lymphocyte? It's only 2, 3, and 4 are correct for B lymphocytes. B lymphocyte bind to specific antigen presented by pathogen, where lymphocyte produce memory cells after encountering a pathogen, leading to a more rapid and effective immune response upon subsequent exposure. B lymphocyte secrete antibodies, which are protein that target and neutralize pathogen. The answer will be 2, 3, and 4. Number 39. Which statement about uses of monoclonal antibodies are correct? Monoclonal antibodies can be injected into patients to give active immunity. Monoclonal antibodies can be injected into patients to treat viral infection. And number 3, monoclonal antibodies can be used as diagnostic test for specific pathogen. The statement 3 is correct. Monoclonal antibodies can indeed be used as diagnostic test for specific pathogen. The other statement 1 and 2 is not correct because number 1, monoclonal antibodies provide passive immunity when injected into patient, not active immunity. Number 2, monoclonal antibodies are not typically used to treat viral infection directly. They are more commonly used for targeting specific cells or protein which involve in the disease processes. Number 14, which statement explains how a vaccination program can control the spread of an infectious disease in a population? Statement B is correct explanation because when a significant portion of the population is vaccinated against a contagious disease, unvaccinated individuals are less likely to encounter infected individuals because the spread of the disease is reduced within the population due to herd immunity. This helps control the spread of the infection diseases. That's all for today's presentation regarding the past year exam paper for 2023. And hope that you subscribe to Jom Study Lab YouTube channel. And thank you for your support and cooperation. And hope that you are doing well in your study. and upcoming examination. Thank you very much.